Hi, everyone. Thank you for watching our final presentation. My name is Nila Murali.
And I'm Vidhya Mallikarjunan.
Today, we will be presenting on possible vaccine solutions for the Ebola and Marburg virus. Before we jump into the specific vaccine technologies and the nitty-gritty of the manufacturing process, first a little background on the importance of this research and Ebola and Marburg in general. Ebola and Marburg are highly infectious and fatal diseases that often present patients with fevers, fatigue, hemorrhaging, and eventually multi-organ dysfunction. There have been over 20 known outbreaks, with a majority concentrated in West Africa. The image on the right shows the regions within West Africa and the years of each outbreak. Although Ebola is so fatal and infectious, there are currently no approved therapeutics for EBD or MBD due to the virus's small size and highly infectious nature. The viruses themselves are from the filovirus genera and have a characteristic filament-like shape with a single-stranded negative polarity RNA. There's one main protective antigen on the virus, a surface glycoprotein, GP. This image shows a magnified view of the glycoprotein. The GP plays an important role for the virus's entry into the host cells and is heavily glycosylated to provide the virus with stability, fusion activity, and shielding. This actually increases the virus's ability to infect and cause harm in patients. Moving into the vaccines themselves, a successful vaccine for Ebola and Marburg require the ability to introduce the virus's vital glycoprotein, such that a strong humoral and cellular immune response is induced with a low immunization and prolonged period of immunity being maintained within the patient. The overall objective of this research is to compare the biomanufacturing processes for this virus and determine which vaccine option would allow for good upstream and downstream separations such that an effective, rapid, and economical vaccine is created. This image shows the considered vaccine technologies for Ebola and Marburg: viral vectors, nucleic acids, whole viruses, and protein subunit vaccines. We investigated similar vaccines as pharmaceutical companies did during the COVID-19 pandemic. The first vaccine type investigated was virus vaccines. There are two types commonly used: live attenuated, which is a weakened form of the vaccine. For example, measles vaccines fall in this category, and inactivated live virus vaccines, which involves chemical or heat denaturing of the virus's genetic material. An example is the hepatitis A vaccine. Both of these virus vaccine types fall within having the same upstream and downstream manufacturing processes. The initial step of cell growth and purification are boxed in red in the manufacturing process description above. The first step to virus vaccine manufacturing is the cell line growth of the Ebola and Marburg viruses. This involves large-scale cultivation of viral strains in suspension. Some of the possible cell lines that can be used are monkey kidney cells and human diploid cells to get post-translational modifications that mimic the virus's activities in humans. The next step is isolation of the virus from the cell culture media and debris using some sort of centrifugation or ultrafiltration to separate any of the microcarriers, cell debris, or cell culture media. Because viruses themselves are relatively large when comparing them to the media, we are able to get fairly high yields. We should be able to isolate more than 2 million plaque-forming units per centimeter squared. The second half of the virus vaccines manufacturing involves inactivation and packaging. The step is weakening or inactivating the virus using some sort of chemical inactivator like formaldehyde or beta-propiolactone to destroy the virus's genetic material. This step can be very expensive due to the cost of the chemical inactivators, and this would be specifically infeasible in underdeveloped regions like West Africa, where such extensive costs would be a little hard to maintain for virus vaccine production. The final step is packaging and transport. Whole virus vaccines need to be maintained in cold temperatures and probably lyophilized as well during storage to maintain their stability. And this is once again infeasible in extreme climates. The main disadvantage of this vaccine is safety. Workers need to continuously encounter live, highly infectious viruses throughout the manufacturing process. And this is specifically important for Ebola and Marburg due to the virus's high infectability rate and also biohazard level of 4. The next vaccine technology studied was recombinant vaccines. This involves a specific subunit of the vaccine, like a polysaccharide or spike protein, being injected into the human to bring about an immune response. Since the Ebola and Marburg vaccine have only one main glycoprotein, that's what's targeted through these recombinant vaccines. One thing to note is that this glycoprotein is heavily glycosylated, which means it also has a lot of N and O linkages, which is going to require heavy upstream glycoengineering and downstream separations. The main step in protein recombinant vaccine production, is cell culture growth. So we need to grow that protein in cell lines. Once again, we want to make sure we get similar post-translational modifications as humans. So we use Chinese hamster ovary cells or human epithelial cells. Then the GP protein is isolated using some sort of chromatography technique or centrifugation technique, for example, metal chromatography or continuous flow centrifugation. These are kind of expensive techniques, and they are required because the protein size is really small. This also actually results in low yields of the protein itself. You only get about 10 to 100 milligrams per liter of the protein per cell culture media. Therefore, the main disadvantage is those expensive downstream processing and time-consuming downstream processing units that are required. And then also, this becomes very infeasible in underdeveloped countries, and how these low yields are also going to require large manufacturing plants in order to kind of go over that solution of low yields.
So the next thing we considered were viral vector vaccines. Viral vector vaccines use the DNA that encodes for a specific protein that elicits the immune response to a disease, adds it to a plasmid backbone that encodes for the outer shell of a virus, and this is because viruses already have the structures needed to infiltrate the body and produce viral proteins. So you can see in this diagram how a viral vector vaccine actually produces an immune response by infecting a cell with the target DNA plasmid and producing the antibodies that will train the body to recognize that later. So EVD vaccines using viral vector technology usually use retrovirus vectors. There's other types of viral vectors that are commonly used in vaccines, like adenoviruses or adeno-associated viruses. But retrovirus vectors have several benefits, including long-term gene expression, which means longer-term immunity, as well as high yields and relatively easy transient transfection, which is the process that inserts the target plasmid into a cell to grow it up. The manufacturing process for viral vector vaccines starts with growing the plasmids in E. coli and then transforming them into human kidney cells, which are called HEK293 cells. The cells containing the vectors are then grown up or expanded and then lysed or broken apart using mechanical or chemical stressors like sonication or chemical detergents or even freeze-thaw cycles. Then purification of the products happens using affinity chromatography because it has pretty high yields and high purity. And then there's fill to finish processing, which we've kind of discussed with all the other vaccines as well. The transient transfection process is the major downside to viral vector vaccines because it's a batch process, and it also requires the use of adherent cell bioreactors, which require materials like roller bottles and flasks, making it really difficult to scale up. There are some suspension alternatives being developed, like Vero cell suspension, but they're pretty novel and, because of their novelty, kind of expensive, which makes it pretty infeasible for target countries for EVD vaccines, like the West African countries we mentioned before. The next thing we considered were nucleotide vaccines, also called RNA vaccines. Nucleotide vaccines inject RNA or sometimes DNA encoding for a target protein to elicit an immune response. They're made by first identifying the plasmid and then transforming it. That's shown in steps 1 and 2 in the diagram. The plasmid is then linearized and then transcribed into mRNA, which is steps 3 and 4 in the diagram. And then the mRNA is prepared for injection. So the diagram shows a specific injection method, but there's actually a couple of different ways to inject mRNA vaccines. There's naked mRNA injection, aka direct injection, where you just put the mRNA directly in, and then there's encapsulation techniques like lipid-based carrier, polymer or peptide encapsulations, or transfection of dendritic cells, which puts the mRNA into a dendritic cell. These are all different methods by which to inject mRNA vaccines. So the downstream bottlenecks in this process are purification of the mRNA vaccines. This is because it's a multiple batch process. It can be pretty difficult, but there are a lot of alternatives. So there are some flow alternatives like SPTFF or aqueous two-phase systems, and they can improve process times, lower costs, but also maintain the high purity. Another alternative is multimodal chromatography. This is still a batch process, but it involves much more intense purification, so you don't need to do the purification step multiple times. And all of these benefits are part of the reason we chose mRNA vaccines to address EVD. There's even more benefits, including the fact that manufacturing systems for mRNA vaccines are highly adaptable, which means it's a lot easier to respond to changes in virus morphology, or even utilize existing equipment from the COVID-19 pandemic and repurpose it for making EVD vaccines. There's also a particle encapsulation technology called MDNP injection, which is really effective for EVD. It allows for a 1-week turnaround for injection-ready vaccines after getting the necessary DNA sequences, and allows for the support of multiple filoviruses. It also, mRNA vaccines also require pretty small bioreactors. Anywhere between 10 and 30 liters can produce enough vaccines for like a pandemic level or like an epidemic level response. And because of that, it's really solid for developing countries like those affected by EVD. EVD is also a really good size. It's about 19 kilobases, which is a lot smaller than COVID, so there shouldn't be any upstream complications. And then mRNA vaccines can also address multiple filoviruses in one vaccine, like I mentioned earlier. And that's super important for Ebola specifically because the regions primarily impacted by Ebola lack robust health infrastructure that's needed for multiple doses of or different vaccines for different filoviruses. All of that being said, it does have a main disadvantage, which is similar and also a disadvantage to viral vectors and virus vaccines, which is cold storage requirements. They have to be kept pretty cold for the mRNA to remain effective. And that's why we suggested adding the ArcTech cold storage device to the national stockpile. So the ArcTech cold storage device basically keeps vaccines cold using just ice packs and its own internal insulation systems. Now that's super important because the alternatives are the Sterling portable freezers and Vaccine Pod portable freezers. Both of those are about $2,000 or even more expensive than the ArcTech cold storage device, and they both also require energy input, which is an even bigger cost in addition per unit. That's just not economically feasible in the target nations for the EVD vaccines. That's why it's so important that we have these ArcTech cold storage devices. They've even been used in the past for serving viral vector Ebola vaccines in West Africa, and they've saved thousands of lives. And so we think it's really important that they be included in the stockpile. Now, if you're interested in doing any more research on the subject, we've included all of our resources that we used in this presentation in our paper. Yeah, thank you so much for watching our final presentation.